and that adult, over when it matures, it would lay the gametes again, and the cycle gets completed. So this is what we are going to cover in the developmental biology. So in these cases, and especially in this course, we are going to study few vertebrates and few invertebrates. Among the vertebrates, we are going to study frog, bird, and fish. And in invertebrates, we are going to study about Drosophila, which is an important model organism that has led us to many discoveries. And little bit, not much about nematode, that is Sonora habitatis elegans. So we'll be learning about these few organisms in animal kingdom, and not only that, we'll be looking at plant also. And especially there is one plant group that we call as angiosperms, and the flowering plants, we will be covering the development, especially early development of the plant, especially the flowering plants and the angiospermic plants. Okay. So not only that, few organs, and especially we'll be dealing with limb development, vertebrate limb development, insect eye, and especially nervous system. Okay. I would like to tell that instead of speaking about insect eye, we will be speaking about eye in general, and how the various parts of the eye gets developed, and how nervous system comes into existence. Then what I mentioned in the beginning, that there would be cause, especially which concerns us, which we are interested and very intrigued to know, how few of the organisms can regenerate their lost organs. Not only limbs, some of them can even regenerate their organs. So how they can do it, and why some of the higher forms of the organisms like humans cannot regenerate their organs, except one or two. None, other organs and limbs cannot be regenerated. So that is the part of the post embryonic development, and one of the aspects of post embryonic development is metamorphosis. So how a completely different adult structure get formed from one type to another, that is from the larval stage to the flying stage, the mostly land dwelling to a free flying organism, how that transformation occurs in insects. And in frog, how a frog transforms itself from a swimming terrestrial aquatic lifestyle to a terrestrial. Even diet changes are there in this organism. How that happens, we'll be covering it under metamorphosis. And of course aging, which is the the important disease of concern for humans. We are always worried about aging, and if you want to pinpoint uh, one of the important targets or goals which humans are trying to seek via understanding of this biological processes, is how to overcome this problem of aging. Aging is the most central problem that we are trying to solve through the knowledge. Yeah, so that is what we are trying to solve. So if you understand the biology in greater detail, we will come up with some kind of a solution to even the problem of aging. So that we are going to understand. So little bit of all these aspects. Of course, the depth would be a matter of time, and restricted to the time. Whatever knowledge we can gain in this area, we'll be doing through the guide that we have in your syllabus. So based on the syllabus and the time we have, we will be covering this topic. Um, so what is interesting about the developmental biology? Because it concerns ourselves, we would be very much eager to know about how our each and every part gets developed. And we have not mentioned in our syllabus anything about humans, but ultimately, from the generalization aspect that is been given to us by the nature, uh, we can study some model organisms and we can extrapolate that information onto humans, and we can seek answers to the problems of humans through these organisms. So they illuminate us with many different aspects and information that we can use. Not only that, uh, we have seen that how we will be seeing actually how Drosophila knowledge has given us a huge window of opportunity to seek answers in the mammals, especially vertebrates. So even though the insect is invertebrate, how whatever we know there has helped us a lot in understanding. So it's an important organism which is very tiny, which we can easily handle in the laboratory, and hence it has become a model organism of choice, and lot of work has been done in this organism. So what we have done is basically, by studying these organisms, have picked up the what we call as the general principles, and we call it as the developmental principles. And we'll highlight few of the general developmental principles in this lecture itself, and we'll try to understand organisms and what general aspects we were speaking in this lecture. We will try to connect it with whenever we will go into any particular organisms later on in the syllabus. Okay. Now there is general principles that we have highlighted that we will be looking at. Before that, we will look at the history of the developmental biology. Now the big question is, what is different about the developmental biology and embryology? So when we look at developmental biology, the time span what I've mentioned is elaborate. We are not restricted to only the embryo part. It is a broad time span we are going to cover. It's like a cycle that we cover, and especially interestingly, what developmental processes are working that we are trying to figure out. But what we have mentioned as a biotechnologist, what we are introducing here is the molecular detail, how molecules, especially the genetic elements and the protein elements, how do they work in a very intricate manner to unravel this developmental program that has been stored in as a genetic information in the in a particular cell, how it is gradually rolled out, and how step by step the processes unravel only because of this specific molecules that get involved at each and every step. Okay. So that molecular detail, when it is picked up and it is elaborated in any information about the developmental processes, then we can safely call it as a developmental biology. So developmental biology has that molecular detail whenever we speak about any development from, suppose we are speaking at the early stage where zygote is getting formed. Okay. And why there is specific areas in the zygote. So we'll always elaborate some molecules. When we go into cleavage, how rapid divisions occur, so what is contributing to that rapid divisions. Okay. So there again we'll elaborate molecules that are specific there. Okay. And how there is what we are calling as gastrulation process. So there again we'll be speaking about the molecules that are involved. So whenever we speak about molecules, the gene, especially when we are speaking about molecule, it originates from the gene. Some kind of gene is always involved. So whenever we bring in that information, we will be calling it as a developmental biology knowledge related to some progress in the development of that organism. Okay. So therein comes the biotechnologists, molecule and unraveling the details of molecules, how they are involved, and the genetic manipulation is the main task that the biotechnologists perform. So biotechnologists come into the picture because they have this technical knowledge of how to open up the cell, how to reach to the genetic information, how to manipulate them, and seek answers that we are speaking about in this embryology aspect. So there we are coming into picture. So hopefully the question of what the role of biotechnologists are there in the developmental biology is hopefully answered and it is clear to you. So we are contributing to the molecular details that is supporting this framework of knowledge of the embryology, and we will then call it as a developmental biology.